think we're well known within the industry, even within academia, but the general consumer would not necessarily know. If you had a yogurt this morning, there's a good chance that that's made with Novonesis technology. The innovation model from startup to scale is very well laid out in pharmaceuticals. So scaling Biosolutions is a costly affair. We actually have examples where we collaborate with some that had a unique idea, but would suffer in the scaling of that.
Do you feel Biosolution is a neglected market compared to pharmaceuticals?
We are actually sensing a lot higher interest for Biosolutions as a key enabler of a greener transition. So we are only starting.
Bienvenue to a new episode. I'm your host Philip, and on the Flot.bio show, I'm interviewing the best Europeans in biotech to help you grow. Biosolutions, sometimes also called industrial biotech or synthetic biology, Synbio, are a major part of the biotech industry alongside therapeutics. Novonesis is the largest biosolutions company in the world. So I went to Copenhagen to talk with its CSO, Claus. I didn't know him personally, but many people recommended him to me, and I've actually tried to make this interview happen for the past 2 years. We discussed the $12 billion merger between Novozymes and Chr Hansen, which led to the formation of Novonesis. We also talked about the challenges that biosolutions startups face and why it really matters to keep a childish excitement for technology. So here's my conversation with Claus, which was actually the first time on this podcast that we've recorded in a studio. If you're enjoying it, please hit the like and subscribe button. Welcome to the show, Claus.
Thank you.
Studio.
Yes, we are becoming a big company in Novonesis, and yes, we do have facilities.
Amazing. So to say, I told you it's the first time I record in a studio like this.
Well, let's make the best of it.
Yeah, let's do it. So I want to start with how we got connected. Keith, so the chairman of Novonesis, helped us.
Okay. Yep. I saw that.
And actually met him in 2015 when he was CEO of Chr Hansen.
Right.
I recorded a video with him actually, and now Chr Hansen merged with Novozymes. I think it was a $12 billion merger.
Yes.
Became Novonesis.
Yes.
I'm curious, maybe you can start there. How did it go, and what did it enable?
Sure. No, and maybe bring a little history to it, because it's not the sort of first time it's been thought about combining the two companies from let's say a research platform technology perspective. Similar, not identical, but similar, and with overlap. And from a market perspective, very different. So we're operating in different markets. So we actually had collaborations before, we'll say, the merger, but then came the opportunity, and I think it was, and we grabbed it.
What was the opportunity specifically?
Sort of a more sort of balanced equity or valuation of the two companies that made it work out, and the fact that we have a strategic owner. Novo Holding helped as well.
Yeah, because they were already owning, I don't know how much, but
they have 25%
of Hansen also.
And then it's been, of course, some preparation getting into the merger, and then execution of what we thought possible on synergies, both leveraging cost synergies, but also, and more importantly, sales synergies, because that's really for the future. That's also where the value really lies in combining such two entities, that we actually gain from having a broader market presence, a better distribution of our sales force, being how do you say, complementary products into the businesses, and we're already seeing that now. Okay. And then, of course, what I'm most excited about from an R&D stance, that's a technology base. That's a technology base. Everything from the repository of strains to what we can do and engineer in those strains, whether it be evolution, natural evolution, or even with technologies like
genetic engineering.
And then of course what these strains, bacteria, fungi are capable of doing and producing.
But I guess there you must have a bit of overlap on the tech stack. With Chr Hansen, I guess.
Yes, but we can fully enable the capacity. This was not about bringing cost down on the research. It was actually to proliferate our pipeline and activities. So we kept the merged cost ratio on research spending, and actually have now programs that addresses you could say problems, customer opportunities from an angle of two different complementary technologies.
That's cool.
So you can start solving for something that wasn't possible with one or the other alone. Does it make sense?
Yeah. And I guess also for the listeners, because the two companies were very close. Yes. Because, I mean, you're both in Copenhagen. I think
10 minutes.
10 minutes from here. So I guess it helps also especially from R&D. I think most of the R&D is here. So the teams are already, are very close together.
Yeah, so the core R&D, what you say is the basic R&D is mainly here. We have as well facilities in China and Japan and US. Yeah. And then applied R&D is something you bring out in the world to the customers. So you're near to the customers with how you apply the technologies in the different businesses.
Yeah. We'll talk about that because I think it's one of the, especially from the Novozymes and Novonesis part, I think one of the specificity of you guys that you work very closely with the customers to do basically custom development of solutions. And I heard this from Estelle from a few people publicly that you are pretty good at this.
Yes. It's very nice to have a very big and efficient and state-of-the-art technology stack.
Yeah,
and we can talk more about that as well. But there's other elements, of course, to being a successful company.
Yes, yeah.
And this piece of what we call customer centricity, nearness to that is, of course, to understand how they operate, what are the key issues in operations, how can we help enable maybe better, more efficient operations, what are the products doing with the consumers in the end, what the quality parameters, performance parameters, so let's say taste, if it's for cheese, you need to understand that, you need to understand how you arrive at that, then you can do the translational piece of okay, so these are some of the opportunities, issues to solve for, and then you can bring that back into understanding what's the sort of biological nature of that problem, and can you measure it, right? And if you can measure that and make it a biological problem, then we can solve for it. And then, of course, you need to scale it. And you see a lot of
just scale it.
You see a lot of technology companies that have that as a challenge also. And here we, by the combination, we are the biggest in enzymes, in cultures, in microorganisms, probiotics in terms of production, both capacity but also footprint around the globe.
I want to unwrap a few things you said, but the last thing you just said, I think what's amazing, Novonesis, you're, I think, now the largest biosolutions or industrial biotech company, I think even worldwide, because you're only doing this, which is pretty amazing. We are proud of our both our heritage and the technology base, what we do, and then being, as we say, a pure play biosolutions company, and having biology as a premise of how we produce and what we produce. And how do you compare to, let's say, a large large chemical company who has a biosolutions part, let's say a BASF or DuPont? I guess they all have a biosolutions part as well.
They have a mix of technologies in. But we leverage that in the sense that's what we do. That's what we do best. This is where we also invest to continue to lead.
But so you guys are bigger than their biosolutions department,
Yes.
basically. Yeah. Okay. Wow.
Yeah.
That's good. And we will talk also about, a lot of things you can help. I think the first thing I also want to talk about is what you said about the end customer, because I think what's really cool with you guys, especially on the podcast, that most people have touched somehow, not touched a Novonesis product, but touched a product that has a Novonesis ingredient or component in it, and often without knowing it.
Yes. Yeah, that's a little bit, that in that sense, we are not that well known, right? I think we are well known within the industry, even within academia, as in the science part, of course, with our customers and so on. But the general consumer would not necessarily know. What they would know is that there's cultures in yogurt, for example. That
they know that the skier is high protein.
They know, and they know that it needs that for cheese making. They might actually have read on the laundry package that it contains enzymes. They may not necessarily know exactly, and they wouldn't know that it was Novonesis producing both the cultures for the cheese or the yogurt or the enzymes for the laundry. But just imagine, so if you had a yogurt this morning, it's probably 50%
here, actually.
Oh, you had it? Okay. There's a good chance that that's made with you could say, Novonesis technology. And the same when you wear a cotton shirt, the polishing, everything that goes into treating the cotton and avoiding chemicals is also enzymes in many cases.
Actually, have you, from a company point of view, have you tried to brand a bit more to consumers, and would it drive business? Or let's say Intel, with the chips, and people know that there's an Intel processor.
Sometimes, especially in the probiotics business, they would, consumers, that there's something to be able for a customer of ours to be branding that it has BB12 or LGG, which are very well-established probiotics. But we only have limited examples where our sort of name is on pack as part of the brand. You remember
because it doesn't add value at the end of the day.
Well, maybe it would to us. We would like to be known, but for our customers, I think it's about their products and their brands. For example, some of in laundry, it's some of the world's largest consumer goods companies like Procter & Gamble and Unilever, and they have their.
They don't want to share the brand too much, I guess.
They have their billion-dollar brands that they protect, and the same with the food companies.
Makes sense. On the application, you mentioned some examples, but actually one thing I was curious preparing was to understand, because I think you're doing quite a lot in terms of application, it's pretty large, but to understand how is the split in terms of business unit. So for my understanding of it, food and health is basically the 45% of the business. I think within that, food is 3/4, human health 1/4, something around there.
Maybe even a little bigger on the food and dairy business. Okay. But you are approximately
somewhere there. And then the second business unit or the second category is more planetary, which I think is 55% of the business around there. And household, that's the historical, the detergent, kind of the origin of the biggest application for Novozymes originally, and then agriculture, energy, which is basically, household 1/3, energy 2/3, somewhere around there.
Yeah, well, energy is somewhat a little smaller than household care, but we did it because some of the technologies overlap into the technical industries. So for example, vegetable oil processing, starch and grain processing, these sorts of things. So we don't have a clean bioenergy reporting. But they are sizable businesses, all of these.
Okay. And can you just talk a bit more about these businesses and what's a key application maybe in in each of the bucket? I mean, we talked about the detergent households.
Sure. So let's do that. But maybe if I can interject a small thing on the first part of this, knowing your customers and their processes and what adds value to them and their customers, if you will. I think sometimes it takes. We sometimes go even beyond that in our innovation. If you know them that well, you can also maybe point to solutions that bring them further than they sometimes imagined.
Yeah,
right? Because you know that. It's like the old Henry Ford, right? Sometimes they will ask for a faster horse, and they will rather bring a car, right? And I think that's the kind of level you need to be at on the application side to really make it matter. And then when we talk about what we then do in the different industries, then of course in let's say our dairy business, we split in cheese and fresh dairy. And some of the latest we are doing, of course, in fresh dairy, is to accommodate for a much higher protein content as consumers demand or wish for, let's say, a more nutritional product. So we develop cultures in that direction. Then it also increases the value of the product. So hence, can you preserve the product? And this biopreservation solutions that we have that prevent mold or mold formation, so it actually also preserves the product of a fresh dairy better. And then again, you can put on top of that probiotic cultures, right? And that again, you said, okay, well, you just put it in. No, you need to make sure that it doesn't do anything to change the profile of the product. So you actually sometimes need to have it, let's say, dormant. So that's just one thing in dairy, and then in the same way you can unfold the different applications, right? And in cheese making, it's of course the whole sort of coagulation setting of the protein in the cheese. To begin with, that's one thing. How much lipids are you able to capture in that process?
And the strains, I guess. Also
and then of course the strains for the fermentation of the cheese. You take household
cheese. I love French.
No, who doesn't love cheese? If you take household care, normally you would say as a consumer, it's about cleaning, right? But for us and our customers, it's also for cleaning at less energy, less water. It might also be for more, let's say, fundamental than just removing spots, but providing a freshness and a renew to the clothes, so you can wear it for longer, and doing it in shorter cycles. So that has a whole sort of benefit for a consumer to have a cleaning experience with much less, cost and effort into it in terms of water and energy. So what do you call it? Cold and quick. So you have. How much do you want me to jump on? Because I can continue.
It's amazing. One thing on what you just said, and I think that's also remarkable in what you're doing. I mean, sometimes I think you're bringing biosolution, replacing something that was done basically petroleum chemistry. I guess into the detergent. That's a typical example. But I guess sometimes in the dairy, you're not replacing, you are bringing something new that was just not possible before, and you need enzymes or probiotics to do it.
Say in the biopreservation, you are replacing, you could say, other preservation agents, right?
But in cheese culture, you would not.
No. There you have to refine it for the taste and texture of the cheese, so the final quality for the consumer, right? So, but then also again, putting speed into the process of the manufacture is you could say that is also an efficiency gain. Increasing yield, so you have less waste. That's an efficiency gain that you could say have a sustainability impact versus the baseline.
And value.
Direct
monetary value.
Yes.
And how much of it's a pretty hard question to exactly know, but how much of the applications are in the category of replacing chemicals or petrochemicals, and how much is more in a...
I wouldn't have exact numbers on that. I do have a number on, let's say, we look at how our products play into the UN sustainability targets. And you can point to 6 of these having relevance in terms of, savings, environmental impact, health, such and so, and nutrition, and these. Okay. And I believe more than 80% of our sales play into, has an enabler. And for most of our customers, it will definitely have for many, most in the processing industry, it's about efficiency gains. And you mentioned household care, taking out polymer, taking out surfactant, adding in enzymes. Of course, there's both a cost out, there's a gain on performance, and there's a sustainability aspect to that. Yeah,
I like that. If we can also zoom out a bit, but talk about biosolution industry, biotech as a field, because what you just mentioned is like you're really delivering value to the customers. But what I'm curious is about more like earlier stage companies, also startups, because on paper it sounds good, but it's very hard to execute. It's also very hard to scale, and especially in Europe, I don't know that many companies that started small, let's say in the 10, 20 last years, and who made it much bigger. So I'm curious a bit on how, and I know you are quite involved also with BII and we had Jens on the show and a few others. How do you look at that?
The thing is, the innovation model from, let's say, startup to scale is very well sort of laid out in pharmaceuticals, yes. Right? You know that well. You can almost sort of put value to the different phasings in the development of a pharmaceutical. That doesn't exist in the same way in biosolutions. But the scalability is, I think the scalability is the hard one. To actually get to high volume product at a cost that is let's say competitive, because to be fair, there's only so much you can demand in terms of a sustainability benefit. You can. It's difficult still to put a value to that, and you would find that many larger customers will gladly take that benefit, but not at a very high extra cost. It has to come. It almost has to come as it's all
added benefit.
And so scaling biosolutions is a costly affair, and sometimes some of these companies have not sort of thought through that process, and maybe they overinvest on something that help them in the beginning rather than seeking maybe the advice and help, let's say, from companies like us on the scaling process. We actually have examples where we collaborate with some that had a unique idea and maybe even the technology solution, but would suffer in the scaling of that. And sometimes we can help with that, and we have other companies that does that. But it's a key parameter. And then getting, of course, the value proposition of the product right. Because many start out from a technology angle. To say many start out of a technology angle. They have the idea, they have the technology, and then they may also have an idea on the application, and then everything it takes from there to putting a product out, and it's surprisingly the goods we consume and use today, how cheap they actually are, right? How much when you buy a product in a supermarket, when you sort of break it down, what was actually the value of, let's say, the biotechnology in there.
But that's an amazing part, actually, because there's two things that comes to my mind. Is one thing is, I think quite a few earlier stage companies think, it's like you can replace petrochemical or something has a huge value. I guess they realize that it's not necessarily the case that you just said. And then it's also very hard to find the application where you are actually adding value compared to what's existing and significant value to capture that value afterwards. Yeah. But what I'm also thinking is that when you know, as you said, when you know the even the R&D budgets or in like industrial scale, let's say in dairy, the margins are pretty low in general. Let's say Lactalis, the margin I think is net profit is whatever, a few percent, 3-4%. So they probably look at cost like very close.
I'm telling you, they look at cost, and you have to be competitive. So if you are talking about exactly, let's say, oleochemistry replacement, it has to come, and you are saying identical. Let's say you want to make a BDO for polymers, and you have to compete head to head with the oleochemical BDO. There might be a niche market that can take a little higher price, but that's a niche market. So the idea of identity is sometimes a hard task for a startup to be competing against, while adding value, doing something unique that couldn't be obtained is a different story, right? That's
so I guess you would recommend startups listening to go more for the unique solutions than the replacement.
There are demands also on food ingredients and others. They become natural, so to say. There are demands for more fermented ingredients. So I wouldn't say never go there and compete, but seek, but be aware that you would have to be cost efficient, and that's about the scalability. It's also about the choice of technology in which you choose to produce, right? And we are very happy with our set production systems, in a sense that they have been evolved quite some more than what you usually find out of an academic setting. In an academic setting, you would often work in cola and yeast, and we are moved to different organisms that has a more efficient in scaling and productivity.
Yeah, because what's the highest batch you do is like whatever 10,000 liter more? What's the biggest?
No, we run up to 200 cubic meters, 160, 200 cubic meters fermentation.
That's really
so they're large. Yeah, and it's not one. It's dozens. Okay. So yes, they're large fermenters.
Yeah.
So
cool. On the capturing value, I want to, after we'll go into R&D on the actual the bioengineering part and go a bit deeper. But first, I want to talk about the value capturing and more about the finance of Novonesis, because what I've seen from a revenue, first it's high, but what struck me even more is the profit or the net profit you get and the margins you manage to get out of it. Which, what was the number I got? Was whatever was, I think for the first 9 months of 2025, I think you had 27% net profit. Somewhere around there, which seems very high.
Yeah.
I mean, it's basically more than Louis Vuitton, LVMH, or even some companies like this. I don't know how, especially given what we just discussed, that it's hard to add value. You're serving industry with very low, quite low margins. So how come, I guess you have unique technology and unique positioning, but how
sure? No. So first, it's of course to have efficient production, and so it sort of boils into what are our gross margins on what we sell versus cost of producing.
Yeah.
We invest quite a bit in keeping cost out of production, innovating also in the way we produce, in the strain that produces it, so gaining productivity gains. So otherwise we would be even more capital intensive. We are, it's not small plants, right? It is capital intensive, but we have been we are able to optimize it year on year, and that's an important contributor to keeping our production cost. Then, of course, we add, our R&D cost. I mean, many of our customers appreciate what we bring in terms of innovation. And that, of course, has a cost, the 10% we invest.
I guess we charge on value, and then
exactly. We have to charge on value for the product. And yes, it's a negotiation, of course, with our customers. We need to be able to show the value. That is, if you're a farmer buying enzymes for nutrition, you can monitor the value, right? And you want to see that. Because here we are also, when you're talking low margins, then talk about agricultural systems, right? And there you simply have to prove it. You have to have, proven trials showing, how much weight gain will I give with this amount of feed for my chickens, and what's the cost of getting that technology that provides that weight gain. And then you can find a way of sharing that. Very much the same in our biofuels business. It's really about the productivity of the biofuels plants, and they can measure the output, they can measure the input, and of course they know the cost on the technology, and we have to show every time that we deliver added value. And that you can then negotiate, right?
And how did the profit margin evolve over the years in the history?
It's been growing.
Okay.
It's been growing. It's not. I mean, we've had in the past high profitability as well, and we have as a target in a strategic period also to increase it further. So our EBITDA, where we project for 25 between 37 and 38, then we have said that towards 2030 we will expand that to 39.
Wow. Okay. Yeah. And then also Chr Hansen was also quite profitable. My sister worked there, and she told me, but it's very profitable.
In that sense, that's another sort of good thing about the simulation.
Business when it's
similar in the way they, we operated and with the financial numbers and so on. It's I must say, it's been quite an amazing journey, I would say.
And I mean, we'll talk about the person, but you have seen it from, I think you joined, 1993 or something?
Oh, yeah.
You have seen the whole journey.
I've seen the journey of Novo Nordisk demerger to become Novozymes, and now the merger, a combination with Chr Hansen to become Novonesis. So tried a lot.
Must be very rewarding to look back and the achievements.
Yes. It's been. It's not. I mean, it's not without bumps on the road, and for which you learn a lot.
We'll talk about the bumps. Well, one thing also about the finance, and this is a good transition also to R&D, is that you're reinvesting a lot. I think what I had the number, I think was 10. Was it 10%? Or was it 10% of sales invested in R&D?
10% of revenue.
Of revenues,
goes back into R&D investments,
which is high. And I guess this is good for you because you have a lot of budget to play with, I guess.
See, that's maybe the difference between academia and a company. Play is probably not the word we would use. No, we have to show a solid return, and that's, then you could say that's my challenge. That these 10% needs to convert to value every year. And we make it that. It comes with the growth, parts of our profitability gains, but that's really sort of on my agenda, that converting that to value and having fun on the way. Because it is rewarding to be in an R&D machine room where you can actually see that what you do actually launches. and we launch more than 30 products a year.
That's awesome.
Yeah, and so if you are an R&D,
it's 30 new products.
Yes.
On top of optimizing the existing ones.
I would say if we replace a next generation, that would count as a launch as well. So it's both the incremental and more transformative. But you can see when you are an R&D, there's a good chance that what you work on will actually go out to a customer. And sometimes you get to see it in a final product that you can buy off the shelf in a supermarket. That's right? Cool. So that's so you could say if that's what I really do and concern myself.
You managed to bring value in the past 6 years.
Sometimes we've been accused of not, right? We had a time. We had a time in Novozymes where we didn't grow as much.
Okay.
In the mid 10s, 16, 17, 18, where we didn't grow, we fell short on our growth targets. And then you kind of wonder, and we were actually running at 12-13% investment in R&D, but it didn't convert to growth. And that's a problem. The problem is actually that we did sell a lot of new products in the period. We had the sales of new products to the tune of 30% of sales coming from new products. But you can imagine what we did was replace a lot of our old. When you have that high, not having growth, you're replacing yourself a lot.
Yeah. Okay.
And if you don't manage to get that, but we did manage to get it at a higher profitability.
Okay.
So you could say we created economic value for our R&D, but this needs to be a balance. There needs to be both a growth aspect and a profit aspect to that. It cannot be only one or the other.
And the 10% is kind of the sweet spot of.
That's where we felt after some time we sort of dialed back in Novozymes. The 10% is a result of the combination. And we feel it's a good place to be. With what we are forecasting, we believe to have a solid return on investment there.
Cool. Let's go more into the science as well. And I've seen that you are. I think, what is it? One of the company, at least in Denmark, with the most patent applications per year, something.
Even in Europe, I think.
Even in Europe, okay.
When it comes in our space.
In your space, yeah. I'm curious on the technology stack of like what you mentioned, but how much of it is like strain optimization, enzyme, how much is it like really into hardcore genetic engineering, bioengineering?
So today you would say most of our enzyme production is done in genetically engineered microorganisms and strains. That's how you get to the productivity, efficiency that allows for some of these applications to exist. Whether it's in biofuels, in household care, wherever it is, even in food. And be mindful that the protein is not a living entity. It's not a genetic modified organism, right? So we produce contained, and then we take the ingredient or enzyme or whatever that strain produced and put it in. And that's as natural as its counterpart found in nature. When you have a protease that degrades protein, it's what we have in our stomach, right, to digest our food. It's the same that another microorganism will be producing out in nature, but we put it into this, let's say, chassis to enhance the productivity. When it comes to cultures and microorganisms, it is evolved from nature by natural evolution. It's selected, you could say, from a strain repository.
To do like high throughput natural selection.
Yes. We have to run several cycles of natural selection. Can we find an offspring, a natural offspring of that? That is better than the mother, right? And so on. So it's like it's like horse breeding on steroids.
But I guess, I was about to say like in even in plants, biotechnology, plants biology, so instead of doing a GMO, they do like very rapid strain selection or plant selection. But I guess in your case, even on the culture side, the iteration cycle is much faster.
Oh, it's much faster. I mean, just the doubling rate of microbial cultures, how they grow and proliferate, is much faster.
So you're not at the same speed as genetic engineering directly, but you're at pretty high speed, I guess.
Yes. No. You will be able to run cycles through the laboratory to find, let's say, a phage-resistant offspring in a culture in a few weeks.
And then you can really target what you want as a property.
Yes.
And you get it pretty rapidly.
But again, this is part of what we do. We also try to understand the mechanisms in which they operate, so we can choose for that. So we can actually put some selective pressure.
Exactly.
We also, when it comes to enzymes, we want to stabilize them, maybe make them more efficient for what they're doing, and so on. So there goes a lot of advanced biotechnology into dealing with all this. This is why we have 10% to understand all this and be able to handle it, and sometimes even develop unique technologies that you could say you can't find or buy to do this. So our toolbox keeps sort of growing, expanding in terms of what we are able of doing. Sorry.
That's amazing. What were some of the coolest patents you've done in the last 12 months or so? Or coolest technologies?
Last 12 months, maybe I wouldn't like to talk about them before they publish.
No, but okay then. So published patents, or maybe the last one, some of them that were published.
I think we've had some very cool patents on some of the biopreservation technology. We've had cool patents on new technologies and a whole sort of new platform for what we call freshness. Of clothes, and sort of this renewing of clothes that gives them so you can use less perfume, have deeper cleaning effects that you would not otherwise have. We have some on new yeast technologies, and now there's a caveat to where you use GMO and not. And when you talk about yeast for biofuels, which is again a contained use of that, then use of advanced yeast, engineered yeast that are capable of simply lifting the productivity to new norms, because and some of the patents in that this is probably also where we have the largest sustainability impact that we can generate.
We'll talk about biofuels also. I mean, just on the science, I'm curious on the genetic engineering of I guess you must be using whatever is available from whatever.
Sure. No,
CRISPR, any NF, CRISPR, multi-gene expression.
No, you could say it goes way beyond. CRISPR is just one tool for gene editing. And you would have multiple tools for gene editing. And then, of course, you have a normal GMO. You take something from a different strain, puts it into this, and then enables this strain to produce that better. So we use all that on the pro. You probably heard about the use of AI, machine learning, to predict structural motifs of proteins. So that is, you could say, that becomes that becomes a new baseline for our research, right? That you have these AlphaFold algorithms, so you can go out and predict structures of, and even maybe identify novel functionalities that you couldn't before. And that's pretty amazing. So you would go out and find completely new families of enzymes. And that happens in a computer today. Bioinformaticians saying, okay, we have this, we know this type of structural motif does that. Let's take instead of searching the sequence, which soon sort of titrates into something that is not the same.
Okay. Yeah.
We can preserve this, look at the structural motif and look at, which other proteins out there will have that structure.
Yeah.
And then you would find something with a lot lower sequence homology that has the same structure and function. That's amazing, right? And that's that we get excited because that means, here's a new wealth of opportunity to bring into play. So that becomes sort of embedded in work cycles in R&D today.
And I guess your genetically engineered strains are like very complex genetic. I mean, I guess it's layers of genetically engineered, whatever, multiplexing several kind of genes.
Yes, both that, but also a part of a natural evolution.
Yeah.
So we do both. So we also continues to improve the strains simply by natural evolution. And then you can say, okay, now sometimes this happens with a lot of changes, and then maybe we can say, we only need this, and this, and then go back and do that. There's other elements to that. It actually creates safer strains. Okay. Microorganisms, you know that there are some bacteria that are good. There's also some that are bad. And many.
So that's the same one, like E. coli.
Yeah, exactly.
Can be good or bad.
Depending on the circumstances. What you can do with many is remove the ability to become bad. Right? So, and that is what we do in our production strains. And that's actually a good thing that you don't bring into, when you do products like enzymes for feed for animals or as a processing aid in food, that let's make sure that's clean. That what we deliver here is absolutely safe and clean. And you can better do that when you're not looking for wild types, strains that may not do it, but it has at least the ability, if not controlled well, to you've heard, of course, of fermentation that goes wrong when people try to make alcohol, right? That is what you don't want.
Like that. And also that reminds me, because I visited the former headquarters from Novozymes, I think was just after 2015 or something. And I remember you had also a huge collection of strains that you were collecting around the world.
Yeah.
How much does that play a role?
But that is still an inspiration of innovation. Yeah. There's two elements to that. One is, of course, this is like a library. You can, but if it's not a warehouse of strain, it's a library. Means that it's ordered. Parts of it is sequenced. You can group it into families. You know what these family does, this and that. So it's really like an index library where you go in and say, I am going to look for this. Let me look in my database on. Do we have? So let's say it was books. I want a book on cars, and I want a book that talks about the fast cars or the engine of cars. And then I can go in and do that. So we don't sort of start from a warehouse and have to pull all the drawers and so on. But that has been an inspiration. But I would also say now from that you can build via if you combine that knowledge with the in silico screening as we talked about before, then you have a really strong. Then you get a really strong repository for new innovation.
And I guess that's where, AI, you mentioned it, we can go a bit deeper. That's where it plays a huge role as well, I guess, because, you have a lot of structured data, and now with advances, and now you even like protein structures, and you can bring everything together.
Yes, it is actually a little interesting that now everybody's talking about it. Data and importance of data and structured data and learning from data and so on. It's a journey we started back in the zeros, right? Because then data became you actually began already at that point of time with the next generation of sequencing, the high throughput screening. That was all in the zeros, right? And that created, a data richness that you had to deal with. So we started, unfortunately, at that time, we had to do it ourselves because you couldn't buy bespoke models to handle all that. So we had to sort of build some of it from scratch. Now, fortunately, you can collaborate with large companies like Benchling or Siemens or whoever. We work with Benchling to do a data repository from end to end. And having, you could say, everything from idea to product linked. And you can, monitor what went on with that. When you have that, then you can start putting your machine learning, and then with machine learning algorithms, AI, on to do more predictiveness, build, let's say, a digital twin for production. Then you can optimize in the digital twin before you actually implement it in the plant. So you can actually move the parameters in silico and say, okay, no, that was not a good idea. Okay, we do this instead. That was better. Okay, and that would narrow, say, the variability that comes out in our production and improve it, right?
And how much the advances in machine learning in the last, 3-5 years and Gen AI, how much does it had an impact on everything you just mentioned?
Yeah, so as I said, we don't do X-ray crystallography anymore for structures. It's AI. It's AlphaFold. And it takes, we're expanding our numbers of structures to more than 20 million over a period of, let's say, in the beginning, it was sort of slowly built up. And we had maybe 6 million last year in the spring. Then we implemented a new service in the NVIDIA GPU, so got a lot more calculation power. And then now it's grown from over a year from a few millions to more than 20.
And AlphaFold, I guess you have a commercial license with DeepMind?
Actually, that some of it is freeware that you can you can actually take, and then you can evolve on that for our purpose. So some of it is freeware. For other things more advanced, it's commercial licenses. But some you can simply it's free.
But you still need to adapt it to your use case.
Oh yes, you always have to iterate a little bit and adapt it. And then the other thing, we would like to keep it our data for us.
I can imagine.
That's usually how companies sees it, right? So our data is our data, and there's an interesting perspective in this, because normally when you do research, you be hypothesizing and looking for a specific outcome, and then you would design your workflow towards that. So you have less interest on the variation around it, and with what we know today, you actually have to create more variability in your experimentation, because that will enrich your algorithm and machine learning.
And the algorithm can manage more complexity.
Exactly. They can easily handle that variation. It actually needs a variation to be trained proper. That's why you see the large language models. They have the whole internet as a data foundation, right? So in the future, some of the capacity that you get from downscaling and more automation, faster high throughput assays, need to be used also to create more variation. So you can,
And I guess also on that topic, the digital twin and the in silico modelization, I guess it's in your case a bit easier than in a pharma setting, where I think modeling the human body is still very complex.
There's a lot of complex. There's a reason why it starts in a place. Now it's a large language model. Where do you have enough that you can teach? There's protein models now. There's a lot, and now with AlphaFold and everything, there's a lot of AI on protein models and the predictability of interaction with molecules, with protein interaction, and so on. So I think in pharma you will see a great benefit of this, especially for antibody receptor or receptor binding and so on. You can start modeling in a whole different way than you were able to. So there, I think, but then the translation, let's say, for a DNA model into productivity, highly complex. Still, also in our world, modeling
like a yeast or strain manufacturing. That's complicated, but I guess.
Oh yes, taking the whole net of if you take a microorganism, it has all the functions you'd say of one of our cells, right? And it really is complicated and can small changes in will make potentially huge changes in the metabolic output of and that is still there's no such thing as a good AI model yet on so even a microorganism. So as you said, doing this on a human body where you have different types of let's say specialized cell functions and having that speak together is even worse. But I do think that pharma will benefit a lot from machine learning because of the variation in data and what we've learned is that some of this will be able to find the odd, interdependencies. I have a story.
A human would not even think about them.
I have a funny story I keep telling is that we hired a mathematician from a insurance company.
Okay.
And he had no idea of biology, right? But he went and looked at parameters from a production of a certain product, and looked at all the variability of what went into this. Records of everything from substrates when you did what to the strain and in the fermentation and this and that. I said, there's no correlation except between this thing. Which I don't know what is, and output here. And then somebody looked at it and says, but that is when we shift dosing profile from a certain carbohydrate to another. And if you do that at that stage, it has detrimental effect. If we do it here, it's better. Good. Let's do it here.
So really data scientists. Looking at it.
It's a small, stupid example maybe, but it just tells you that some of these technologies will take us to new places.
Yeah. We haven't talked too much about biofuels, but maybe in another episode. I want to move a bit more to the a bit personal aspect. What struck me, you seem to have a lot of fun in what you do.
I get excited about what we do. I must say, I also get excited by the fact that it's used for something. Because if you're going to ask me a personal question of why I entered the industry, is because I couldn't see myself in academia.
Okay.
I was there. I started there, but it was slow and. It's okay to try to make sense of things and so on, but it most of it was not very useful. That's just, and I'm not saying that academia is not useful. That's not what I'm saying. It's just
there's no direct application.
No, and I liked to see that. I like to see it, become something that matters. Science at university does that, but it does it as a foundation for which companies like us and others can build, solutions, innovations.
And how did you manage to sustain this enthusiasm and fun over the many years?
It's never status quo in biotech. When I started, I guess some of the first products were out 5 years earlier. The first GMO derived production was in the late 80s, right? The whole idea about protein engineering was almost new. And as I said, then came high throughput. Automation technologies, DNA sequences, the human genome was sequenced in the early zeros, right? And finalized there, and everybody thought it would now revolutionize disease treatment and so on. And it just peeled another layer of the onion. And then now it's about understanding the intercomplexities of whatever SNPs and whatever else happens from a human genome to the translation to the human being, right? So, but it comes in these, and there's always something new. There's things we couldn't do 20 years ago that all of a sudden becomes an opportunity because you can do it now.
And I guess for everything we just discussed, also you're able to take the new technology and really test it, like apply it right away, like what you said with AI, and you can directly see if it translates to value and to value for your customer and for the end user as well. And you can do that at scale, which is, I guess must be pretty...
It is very rewarding. Yeah. And sometimes I feel that our team is like, the top champs, right, in the soccer league of Biosolutions, and that's fun.
That's correct. The last personal question before a bit more quick fire, quick question, quick answers. I think you lived in the US for a bit, and Novonesis is a very global company. How important was that? How it fitted in the picture with your current role?
I think it's important. The idea that you know the world from sitting in Copenhagen is wrong. The idea that it takes realization, the understanding that we are a global company. You get that sense when you go out in the world. When you start understanding that some of our businesses are a lot bigger outside Europe, right? And when you talk about household care, we talk about bioenergy, there's almost nothing in Europe. So what brought me to the US was bioenergy at the time. And you get a sense of different cultures, different work, ways of working. But it's also paramount for learning new technologies, talent attraction. When we started in California, for example, it was to sort of tap in, and that was already back in the early 90s, tap into the whole wealth in molecular biology that took place on the east and west coast of the US. And so
genetic factories, Genentech.
Yes. So you need to have that, and you need to have that, outlook in the world, and you can't look out from Copenhagen. Then you need very strong.
How much you traveled into the different?
Every year I have to go, U.S., China, Japan, and also where customers takes us. But it's not. I would, it depends on the function. I'm okay. It's been worse, I would say, for what it is today. But it's part of it.
And I didn't ask, but how much is China playing a role? Because right now in the pharma world is like it's very. I mean, not just in pharma, but in pharma world it's quite hot that China is growing and bringing a lot of innovation. How much is it true in industrial biotech?
But they also within industrial biotech, they actually are the largest on some fermentation products. So bulk citric acid, vitamins, these sorts of fermentation products, which you would also call biosolutions, but these are more bulk productions.
And a bit commoditized, I guess.
Yes.
In some places.
Yes. We've been in China with production since the early 90s.
Wow.
Yeah. Okay. We've had R&D in China since 90s. We built the R&D headquarter for China in Beijing in 97.
Wow.
Stayed there since. So in that sense, I think we have good sort of relationship with the Chinese innovation scene. In the beginning, it was not very. I'm not being honest here. It has advanced some since the 90s, but it's advanced at a completely different pace than in Europe and the US. To an extent that now you probably have as much activity in China on as you said, pharmaceuticals. Yeah. I would say also in parts of Biosolutions in China, as in many places in Europe and the US. Yeah. So they are advances, and you can start now collecting innovation from the outside in China, collaborating with Chinese Institute for novel technology. Yeah. We also do that, cool. And we can only do that by having a presence and being trusted as an entity in China.
It's cool. Yeah. So quick fire, quick answer, less than 5 minutes. Do you feel biosolution is a neglected market compared to pharmaceuticals?
I wouldn't say neglected. I think we are actually sensing a lot higher interest for biosolutions as a key enabler of a greener transition. And that goes across agricultural systems, food systems, what we do and how we produce. So there's even in the EU a recognition that biosolutions is a thing. Actually, across US, China, Europe, biotechnology has been spelled out as a key critical technology. And that is not only for pharma, diagnostics, vaccines. It's also for biosolutions, for cleaner and more efficient agricultural systems, for the whole food manufacturing systems, for everything you do, right? Yeah. So I'm not saying neglected, but there's a lot more to go for. That I would say. So we are only starting.
More personal question. One mistake you made in the past 12 months?
In the past 12 months? Let's see. They've not been so dramatic. I'm sure. Well, if you take decisions, you take wrong decisions. Let's say that.
It's okay.
I'm sorry, it's not a. I've made bigger mistakes some years back.
An example? If you have one example,
I think the belief that you can sort of. I worked on biomass, so enzymes for converting biomass, not grains, but waste to fuels. And we kept at it, refining, you would say, the biotechnology around it. Yeah. And not realizing that the problem is elsewhere. It's in the infrastructure collection and treatment prior to our technology coming into play. And we invested then into something at a point of time that we didn't understand well. And that cost a lot of money. And it didn't move us. Now the industry has moved since, because it took engineers, from companies like Raízen in Brazil, you could say a sort of a fuels, chemicals company with heritage in let's say even from the oil business, right? To take and engineer such. Yeah. That's not for Novonesis to do. And if there's something to watch out for, it's also for now. We still look at these more transformative opportunities, and then I have to be careful of dosing the R&D to a level, and then maybe something else needs to happen before we can really. Does it make sense? Yeah. Because a wrong decision would then be to continue to hammer at something that you are not in control of when what you have is good enough. Yeah.
Last one. One advice to 40 years old Claus.
So I'm older than 40, right? So I'm going back in time. Exactly, I'm going back 18 years in time. To 40-year-old Claus, I would say. Keep your sort of a childish love for what technology can do. Yeah. But maybe focus as much on the people that are doing it. I don't know if that makes sense to you.
Totally.
But because you can get, all sort of involved with the outcome, the impact, the what the technology can do. But maybe it's as much about how people. Yes, but yeah, seeking, the talent. You often you would, as I said, replace a molecular biologist with a molecular biologist, right? And that's sort of. But sometimes you need to go in different ways to get something new. That means nursing, also odd. Ways of looking at things a little more than I maybe have done, because that would maybe have moved some of our technologies even further.
I like it. I think you have kept the childish. I can feel the childish love for technology. I can feel it. Great. Thanks, Claus. Amazing conversation.
Thank you.
Thanks a lot. I'm impressed by how Claus is so enthusiastic about the technology and product he developed, even after doing it for over 30 years. I'm also impressed by the impressive financial results of Novonesis that he contributed to. If you enjoyed this episode, please hit the like, subscribe, or review button. Any of these actions help many more people discover the podcast. We also have plenty of similar videos on our channel, so please feel free to check it out. I would also love to hear what you think, so if you could leave a comment wherever you are, or shoot me an email at philip@Flot.bio. That's philip@flot.bio. All right, thanks so much for watching, and see you in the next episode.